Good morning, students. Hello, good morning, students. Can you hear me?
Yes, sir.
Yeah, good morning. Okay. So I think last time we discussed about uh, bacteria having linear or multipartite genome, and I remember discussing about plasmids as well, right? I think we saw this image of plasmids and we discussed that there are different types of plasmids, correct? Yes or no? Can someone tell me? Okay, fine. So basically there are these 5 types of plasmid. You have resistance plasmid. Resistance plasmids are those plasmids which encode for antibiotic resistance. Okay. So whatever antibiotic resistance gene normally the organisms have, most of them you will find them on uh, the plasmid itself. Okay, in very few cases, you will see that this resistance may be encoded by the genes on the genome of the organism. Okay. Now, fertility plasmids are basically those plasmids which code for the pili, which is responsible for bringing about a process called as conjugation, wherein they can share the plasmids, right? So that bridge is formed by a proteinaceous structure, which is called as pili, and only the organisms which have the pili will be able to carry out the process of conjugation and share their genetic material. And that's the basic reason why it is also referred to as fertility plasmid, as you can see. Other than this, there are killer plasmids wherein they synthesize chemical or toxins to kill other competing bacteria. Okay, so we call them as killer plasmids because they are encoding for chemicals like bacteriocins so that the competing bacteria can be destroyed. They can be killed. There are also degradative plasmids. Now, what are degradative plasmids? These are those plasmids which code for those enzymes which can bring about the metabolism of those materials which are difficult to be biodegraded, right? Like you can see metabolism of plastic. It's difficult normally. Plastic degradation by itself is a very difficult process, but there are organisms which have the ability to bring about the degradation of plasmid. Okay. And there that ability is degradation of the plastic which will be encoded for in the plasmid. So such plasmids which will be referred to as degradative plasmids. You can also see that there are organisms which have the ability to degrade some aromatic hydrocarbons as well, which is normally very difficult to break down, and it may take years together to break down that particular chemical. Okay, so these plasmids will be called as degradative plasmids. Now, there are also plasmids which encode for pathogenicity. So they are called as virulence plasmid. Best example for this could be the TI plasmid of Agrobacterium tumefaciens, wherein you will see that their ability to induce the crown gall disease. You would have seen that in several plants, especially the dicotyledonous plants, on the leaves you see these red colored tumor-like structures. Okay, that is the crown gall disease, and it is caused by a bacterium called as Agrobacterium tumefaciens, and the virulence that they have to induce this kind of an infection in the plants is encoded for in a plasmid, which is very popularly called as the TI plasmid. Okay, now let us move ahead and try and understand what are the other features about the bacterial genome that we need to know. Okay. Now, with a bacterium like E. coli K12, K12 is a strain of E. coli. Its chromosome is around 4.64 megabase pairs. Okay, and it can also have a variety of plasmids within them. Okay, so along with the genetic material, along with their chromosome, they are going to have different combination of plasmids, and they could be, few kilobases in size. Those plasmids could be few kilobases in size, and you have to understand that these plasmids are going to be extra chromosomal material other than the chromosome. So chromosome ke saath saath they could have variety of plasmids, and let me tell you that the number of plasmids could go in few hundreds to few thousands as well. Okay, so it is going to depend on the organism, on that particular strain, as to how many different plasmids does it harbor. Vibrio cholerae, O1L tor. O1L tor is the type of strain of Vibrio cholerae. This is a pathogenic bacteria again which causes cholera, a waterborne infection. It has two circular DNA molecules, so it's a multipartite genome. I have already told you what multipartite genome is. It's basically wherein the genome is divided into multiple parts. Okay, so here you can see that their chromosome is divided into two molecules. One is of 2.96 megabase pairs, while another is of 1.07 MB, right? So 73% of the organism's genes, total genes, which is around 4,113 genes, will appear in, the DNA molecule which is of larger size. That is the 2.96 MB one that contains the maximum number of genes. Okay, while 1.07 will have the rest of the part of the genome. Okay, now you have to understand that this doesn't mean that एक DNA molecule के साथ उसका काम चल जाएगा. जब भी ये बैक्टीरिया डिवाइड करेगा, उसको दोनों ही DNA molecules के कॉपीज जो है वो बच्चों में बांटने पड़ेंगे. एक मॉलिक्यूल के साथ उसका काम चलने वाला नहीं है, भले ही उसमें 73% of the genes क्यों ना हो, लेकिन few very significant genes may be present in the second DNA molecule. So without the second DNA molecule, that organism is surely not going to be complete, and it may even affect its survival. So you have to understand, bada ho ya chota ho, DNA has its significance, and if it is present in multiple molecules, then it has to be, all those molecules has to be passed on to the future generation for them to possess those characteristics as the parental characteristic. Okay. Now, please understand that if you see Vibrio cholerae's case, a closer examination on the type of the genes that are there in both of these DNA molecules, you will understand that the proteins, important proteins, or energy generation based enzymes, which is your metabolic enzymes, or pathogenicity and virulence based enzyme, most of them are located on the larger molecule. Because it's obvious that you are already saying here 73% of the genes are present in the larger molecule. So most of the important proteins you will see are coded for by the larger DNA molecule over here. Okay, the smaller molecule still contains essential genes, as I said. Okay, and you can see that they are having certain important characteristics which plasmids exhibit, like a presence of integron. Okay, a set of genes, that has an origin from bacteriophages or other plasmids, which may not be originally the characteristic genes of Vibrio cholerae, but it has come from the other organisms, or like, bacteriophages. Bacteriophages are those viruses which infect the bacteria, or even maybe from the other plasmids. So the smaller DNA molecule, which is present in the L-tor strain, it basically tells you that it is not something which is a classical set of genes which this organism had. It is a combination of genes which has come from other organisms, and there is a presence of integron, which basically is a type of a genetic mechanism which is going to help the organism to adapt and evolve very quickly by expressing new genes. Okay, so this is an indication that this is a very adaptable strain, and that is also risky. It is risky from the point of view of the treatment strategies, the therapeutic strategies, because it's a clear-cut indication that because this organism has an ability to evolve faster and adapt faster, there is a higher risk of it gaining quicker resistance to the antibiotics that are currently in use. Okay, so that is something that we all need to keep in mind when we talk about the L-tor strain of Vibrio cholerae. Okay, so the smaller DNA molecule is just like a plasmid here. They are referring to it as a mega plasmid because it's slightly bigger than what a plasmid would be, but its characteristics are, very similar to plasmids, and somehow, the genes that are there on the smaller genome, it has been obtained by the ancestors of the Eltor species during the process of evolution. Okay. Also, there is another very interesting organism called as Deinococcus radiodurans R1. R1 is the strain again. So radiodurans is basically a bacteria which is resistant to radiation. Normally, when you expose any bacteria to radiation, it is going to show you cytotoxic effects, and you will see that the organism will not survive for long. Okay, but the case with radiodurans is that they have many genes which help this bacteria to resist the harmful effects of radiation. Okay, and obviously you will see that here also their genome is multipartite, wherein they have two circular chromosomes, and along with those circular chromosomes they also show the presence of two plasmids. Okay, now, still Vibrio and Deinococcus genomes, we would say that they are not very complex as compared to what Borrelia burgdorferi's, genome is, because this particular bacteria is the one which is responsible for causing Lyme disease, which is very common in America, especially North America. You will see that Lyme disease is very common. It is spread by a vector called as ticks. Okay. Now, their genome is a linear chromosome of 111 kilobase pairs, and it has around 875 genes. Okay, but along with their chromosome, see, they have 19 linear and circular plasmids. Now, 19 is a huge number because they are not just copies; they are distinct plasmid molecules, which means that every plasmid is having a different gene there, different set of genes there. So they contribute to another 504 kilobase pair. So क्रोमोसोम 911 kb का है, और यहाँ पे 504 kb जो है वो 19 प्लाज्मिड्स मिला के आ रहा है, which contributes to around 478 genes. So obviously this is much more complicated than what the Vibrio and Deinococcus genomes are, right? Now, although very little has been known about the function of these genes because they have not been characterized to a great extent, but surely, there have been several genes which have been recognized for contributing for forming membrane proteins, for synthesis of nitrogenous bases like purines. Okay, so several genes have been identified, and yet several are, their function has to be assigned as yet. Okay. Now, one thing is for sure that the Borrelia plasmids, they are essential components because this is for sure that prokaryotes, which show highly multipartite genomes where the genomes are broken into several pieces, into several molecules, and there are multiple number of DNA molecules inside the organism. So this Borrelia is one of the best study model organism that you can have to study multipartite genomes, because as you can see here, the linear chromosome is 911 kb, and then you have 19 linear and circular plasmids. So it becomes the best model organism to study multipartite genomes, and their composition obviously is very different from what a eukaryotic nucleus looks like, because a prokaryotic arrangement is always quite different from a eukaryotic nuclear arrangement. So Borrelia can be considered as a model organism here to study the concept of multipartite genome, because yet a lot is left behind there to be designated in terms of the functions of the genes. So here you can see this table in the book of T.A. Brown tells you the examples of genomic organization in different prokaryotes. It's basically speaking about all four organisms that we spoke of, wherein you can see that the size of the genome and the number of genes and the number of DNA molecules has been written. So you can see K12 E. coli has one circular molecule of 4.642 MB, and there are 4,315 genes which are present. And in Vibrio cholerae Eltor, you can see there is a chromosome and there is a megaplasmid, and you can see the sizes are different and the number of genes are also written over there. Same goes for Deinococcus and further Borrelia as well. So they have told you the genome size as well as the number of genes which are present on each of these molecules. Now, at a later point of time, we will also be trying to establish if there is any link between the genome size and the number of genes. यानी जितना साइज होगा उतना नंबर ऑफ जीन्स होगा, ऐसा कोई रिलेशनशिप है क्या, ये हम एट अ लेटर पॉइंट ऑफ टाइम इन द डिस्कशन ऑफ प्रोकैरियोटिक जीनोम एस्टैब्लिश करने की कोशिश करेंगे. But right now, what we are interested in is understanding few terminologies. Okay, so after seeing Vibrio and Deinococcus, it was necessary to come up with some terminologies to distinguish the chromosome, plasmid, and also, there is another term which is coined now, which is called chromid. Okay, so let us see what these basically mean. Now, chromosome is the actual genetic material of any bacteria. Okay, there could be a single molecule of that chromosome, or it could be broken down into multiple pieces, making it a multipartite genome. Chromosomes are known to carry essential genes, and they are located in the nucleoid. Okay, they are responsible for most of the essential genes for survival of this organism, and is organized in the form of a nucleoid, which we have already discussed earlier. Right? We have seen that there are some proteins which are very similar to histone, while there are some proteins which are very different from what a histone-based organization is. So we have already discussed about the concept of nucleoid in this particular scenario. The second term is plasmids. These are called genuine plasmids. Now, these are obviously different from what the chromosome is, okay, and they are a separate molecule. And these are those genes which give special characteristics for the bacterium, but they consist of non-essential genes. Now, please understand what a non-essential gene is. Essential genes are those which are going to be responsible for coding for those proteins which are essential for the survival of this organism. Which means that अगर वो gene नहीं रहा, तो इस organism के लिए survive करना मुश्किल हो जाएगा. Non-essential genes are those which code for products which are not essential for survival. They may endo some additional features, additional characteristic to the organism, like antibiotic resistance or some degradation ability, which may not be directly linked to the survival of the organism. So plasmids are those, genuine plasmids are those जिसपे non-essential genes होते हैं, and they are distinct from what the chromosome of that organism is. While chromids are a type of plasmids, but they are carrying essential genes. So they are carrying those genes which are important for the survival of the organism. So such plasmids which carry essential genes are what we are referring to as chromids, which is a combination of chromosome and plasmid. So you call it as chromids. So according to this nomenclature, according to these terminologies that we have discussed, V color A has one chromosome, one chromid. And radiodurans has two chromosome and two chromids, which means that the plasmids that they are possessing, they are carrying essential genes. So hence we are not calling them as genuine plasmids, rather we are referring to them as chromids. Is the terminology clear to all of you? Can I see a yes in the chat box, or at least unmute and speak? Have you understood the concept of chromosome, genuine plasmid, and chromid? There are 21 people in the class. Great. Okay. Let's go ahead then. So this image is basically telling you the definition. The figure has nothing to describe rather than just showing the variations in size. But the definition is given over here, that the chromosome is located in nucleoid, carries essential genes. Chromid uses plasmid partitioning system, carries essential genes. Plasmid uses plasmid partitioning system and carries non-essential genes. Plasmid partitioning system is nothing but it having its own replication mechanism in place. Okay. Now let's go to the genetic features of the prokaryotic genome. So let me tell you that it is very easy to annotate genes in the prokaryotic genome by just going through its sequence. You can recognize different genes that are there by comparing it with the databases that are available, right? Bioinformatics tools are very significant for this purpose, and most of the prokaryotic genomes have been sequenced now. Okay, so we know most of the genes that are present in prokaryotes commonly, so it becomes very easy to recognize that which are the genes that may be present on the basis of the sequence that you have got by carrying out genome sequencing. Okay, so this is how you can also estimate the number of genes that are present and also have an idea of what are the functions that the genome of this organism can carry out. But let me tell you that many a times there may be genes which go unnoticed, which we are not aware of till date, and you are missing them out in terms of recognizing them as a gene and recognizing their function. Okay, so there have been several surprising results which have come out in the study of the genome, which has forced microbiologists to reconsider the meaning of species when it is being used in terms of prokaryotes. Because there are lots of similarities across species also, which makes the classification process a little difficult in prokaryotic organisms. In eukaryotes, it is still a simple process. The only thing is that in eukaryotes you have a specific classification pattern, but in bacteria that rigid classification pattern is very difficult to be applied, because there are so strong similarities between organisms, even though they may have some distinct and unique features. So classifying them becomes a big task. Now let us understand the genome organization that is there in E. coli K12 genome. Okay, so bacterial genome, they are very compact because the bacteria wants to package as much as the genetic material as possible in a very small cell. So they have a very compact genome, and you will see that where there is very little space between the genes. In eukaryotes, that's not the case. In eukaryotes, you will see that there is a lot of space between the genes. Okay, but that may not be the case with prokaryotes at all. So we need to understand that when you talk about the genes which are present in the prokaryotic genome, you may not find that there are gaps between two different genes. They may be very closely packed with each other. So when you scan the open reading frame, okay, you should have this point in mind when you're identifying the genes in the genomic sequence, that some of the genes may also end up overlapping with each other, which means that in one gene, you may have the second gene already starting there within that first gene. So you could have overlapping sequences also. Okay, so this kind of a DNA is what we call as intergenic DNA. Okay, and you can see that when you map the genome, you will be able to recognize that there are multiple genes which are even overlapping, and there is around what you call as 11% of the total DNA which you will find in this overlapped fashion. Now, E. coli is one of the model organisms for prokaryotes. Okay, whenever you speak about prokaryotic organism or bacteria, the first name that comes to mind is Escherichia coli. And E. coli is first of those prokaryotes also whose genome was completely sequenced. Okay, and it has been the standard for us to compare it with other known bacterial species whose genome were sequenced, and we wanted to identify what their role is or what their function is. Now, compact organization of the genes is very beneficial to prokaryotes, because even with such a little amount of space, they are able to have multiple types of genes, which means multiple types of functions. Okay, and unfortunately, studying this becomes a difficult task, and providing an experimental evidence also becomes a slightly difficult task. Okay, and but this is surely known that the number of proteins that they express is really high in number, even though their genome size is comparatively smaller. Normally, तुम जब eukaryotic genome देखोगे तो उसका size बहुत ही बड़ा है as compared to what a prokaryotic genome is, and that makes it easy for you to relate that okay, इतना बड़ा genome है तो इतने ज़्यादा proteins भी बनेंगे. बट अगर तुम प्रोकैरियोटिक जीनोम देखोगे तो वही हमको सरप्राइज करता है कि भाई जीनोम इतना छोटा होने के बावजूद भी there are multiple different proteins that are being expressed by the genome. So you can see that this is the genome of E. coli K12, which is being shown over here, and at the top with the green arrow, they have shown you the origin of replication, where the replication process begins. You can see that it is 4642 kilobase pairs that has been written over here, which is the size of that genome. Right? So they have shown you that there is a strand which you can see outside, and there is another strand which is being shown you inside. So genes on the outside of the circle, which are indicated as red in color, they are normally transcribed in the clockwise direction, while the genes which are internally shown in that orangish color is the one which gets transcribed in the counterclockwise, that's the anti-clockwise direction. So this is of E. coli K12. Okay, you can have such a genome map of other organisms also and other strains of E. coli also, which will be available in several of these bioinformatics softwares or databases that are there out. Now, this is again a 50 kb segment of the E. coli genome that has been shown to us, okay, where you can see that they have shown us a specific fragment of the DNA and not the entire genome as the whole. And what you have to note over here is that the genes are quite close. If you can see, I'll just use a pointer to show that how closely the genes are packed. So you can see that, look at this, that the DNA is, the genes for coding for these particular proteins are very closely packed. Here also you can see that the genes are very closely packed. While in some cases, you can see that the genes are overlapped. Can you see here that two proteins are being indicated coming from the same segment, which means that the genes are overlapped. Here also you can see that the genes are overlapped. So this tells us that how the prokaryotic genome, even after being short in size, has the ability to express a variety of different proteins, because first the genes are very tightly packed, and second the genes are overlapping. Now, when you compare this prokaryotic genome segment with that of the human genome, you will automatically realize that the number of genes which are there in the E. coli segment are higher in number, even though the space or the size of the entire genome is short, but they end up packing a lot of genes by reducing the space between the genes and bringing about overlapping of genes. Okay. Some genes virtually will have no space at all, or they are completely overlapping with each other, as we have seen in the earlier case, or in some cases they may be separated by just a single nucleotide in between. Okay, or they may immediately begin also. That is also possible. Okay, please understand that such closely packed genes, they become an example of operon. Now, what is an operon? So operon, since a very long period of time, has been considered to be a characteristic feature of prokaryotes, though there are a lot of reports coming in that operon-based models or similar models could also be reported in eukaryotic organisms like yeast, at least. But it's a characteristic feature that is very evidently recorded in prokaryotes, wherein a group of genes which are involved in the same pathway, they are closely packed as a cluster. So all of those genes are involved in the same process. Like this THR-ABC, all three genes are involved in synthesis of amino acid threonine. So you can see that they have been packed very closely with each other. Okay, so it becomes like this area on the genome has become the section for dealing with the synthesis of amino acid threonine. Okay, so several such operon models you will be able to see. We are going to take two examples at the end of our discussion of prokaryotic genome, wherein we will see what operon is in more detail. But right now, what you have to understand is that they are a group of genes which are involved in a single biochemical process, and they are expressed also in conjunction, that is together or one after the other, and they are packed very close to each other. Okay. Now, in some cases, the genes belonging to the operon may not be clustered together, but may be present at some distance. But then to bring these genes close together, the DNA segments might loop. They may fold themselves in such a way that the genes are brought in close contact with each other. So it is not at all necessary that they have to have a physical proximity. In some cases, they may be separated by few genes or few nucleotides, but then they are brought close to each other by looping of the DNA. Okay. Now, bacterial genes, they normally appear slightly longer than the archibacterial genes. Archibacterial genes have been found to be comparatively shorter. Okay. Now, in prokaryotic genome, you will see that repetitive sequences are less, which in eukaryotic genome you will see in higher number, that repetitive sequences are more. But infrequency of repetitive sequences is seen in prokaryotic genome. Okay. Now, they do not have, repeat sequences, which we call as interspersed repeat sequences, which are normally seen in eukaryotic genome. They don't have such high copy number of repeat families in their genomes. They surely possess certain sequences that are repeated elsewhere in the genome, which are called insertion sequences, like insertion sequence 1, insertion sequence 186, which is around 50 kb segment. But again, their repetition is not as high as what you find in case of repetitive sequences in eukaryotic genome. The repetitive sequences in all are less. These insertion sequences, these are example of transposable elements. You have heard about transposons from your junior college itself. You call them as jumping genes. These are the sequences which have the ability to move around the genome. Okay. And also these insertion elements, they have the ability to transfer from one organism to the other. Even sometimes this transfer may occur between completely two different species. So it is not at all necessary that if an insertion sequence is present in E. coli K12, it has to get transferred in E. coli itself or E. coli K12 itself. It may transfer into a completely different bacterial species also. Okay, that is also possible. Now, the position of these insertion sequences, they tell you that which isolate are you actually studying, because their positions normally are indicative of the isolate that you are working with. Okay, but in different isolates, different species, these insertion sequences may be present at different positions. And in some bacterial species, they may be completely absent also. It is not at all compulsory that every bacteria needs to have an insertion sequence. No. It's not at all necessary that every bacteria needs to have a transposable element. Not at all. But if it is present, their position may be indicative of the isolate that you are studying or you are working with. Okay. Now, many prokaryotic genome, they have at least a few non-transposable repeat sequences. Transposable repeat sequences to humne dekh liya, but most of the genome you will see that have few non-transposable repeat sequences, and they have been classified into different types. You have repetitive extragenic palindromic sequences. These are 20 to 35 base pairs in length. They may occur either singly or they may be present in arrays. Okay, and these are transcribed into short RNA molecules. These RNA molecules that are formed, they can form stem loop structures. And basic role that these sequences have, that is in gene regulation, because these RNA molecules will interfere with the transcription translation process of specific genes, bringing about the regulation of protein expression. So they are not just waste repetitive sequences that are present there. They have a role to play, that is to bring about the regulation of the expression of specific genes. And then you have those repeats which are hyped these days, which is clustered regularly interspaced short palindromic repeats, which are also called as CRISPRs. Okay. Now, this basically is a part of programmable nuclease. Okay. And its role again is in regulation. Please understand that here their role basically is going to be very, very important to inactivate the phage DNA. It's a part of bacterial immunity. Okay, it is trying to inactivate the phage DNA, which is trying to infect that particular bacteria. So CRISPRs have been evolved in order to defeat the infection mechanism or the infection process of the bacteriophage. So these are 20 to 50 base pair sequences, again found in tandem arrays. These repeats may be separated by spacer sequences of similar length and a unique sequence of that spacer sequence. These spacer sequences, which separate these repeats, they may resemble the segments of bacteriophage genome. They may look very, very similar to what a bacteriophage genome is. And let me tell you that the transcripts, the RNA molecules which are formed from these spacers, they are going to act as guide RNAs. Why guide RNAs? Because they will let the entire system understand which part is the phage genome, because it's going to be the complementary sequence of the phage genome. Because as I said, the spacer sequence, see, I'll just write and show it to you. That will make more sense. Just give me a minute. So let us say that you have Crispr sequences, and then you have spacer sequences. So Crispr sequences are basically separated by same length of Crispr sequences and a unique sequence. So these are my spacers. Okay. These spacers are very similar to the phage DNA sequence. These spacers, when transcribed, will give me RNA molecules, which I call as guide RNAs, because their sequence is going to be complementary to my phage DNA sequences. So this will help me recognize where is the phage DNA, because as they are complementary, they can go and have a temporary, linkage or hybridization to the phage DNA, which then enables a set of proteins which are called as Cas proteins. Cas or Cas as you call it, these are endonucleases. So जहां पे भी जाके phage DNA तुम्हारे क्रोमोसोम में इंटीग्रेट हुआ होगा, वहां पर जाकर Cas प्रोटीन उसको काट देगा और वहां से उखाड़ के फेंक देगा. बट ये phage DNA एग्जैक्टली कहां इंटीग्रेट हुआ है, ये मुझे कैसे पता चलेगा? ये मुझे पता चलेगा from the guide RNA, which has a complementary sequence to this phage DNA. So it will go and bind and hybridize to that location, and the Cas protein will cleave it off from that site. So this is the technology which today we call as CRISPR-Cas9, which is a very popular gene editing tool. Is it clear till this point to all of you? Can I see some response from you guys if you are here yet? Hello. Others? Okay. Just give me a minute. So I'm going to show you a quick video for the CRISPR-Cas9 technology. Let's have a look at that, and then we will go ahead. So once you're able to see and hear, I want at least few of you to type yes, so that I know that it's audible to you guys, okay?
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The Crispr method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so I hope that that video has thrown some clarity to what the technology of CRISPR-Cas is all about. Okay, now there are sections of operons. Obviously, after a little discussion more about some more parts of the repeat sequences, we will be moving to operons, but that's hardly like a little part of it. And after operons, we will be trying to establish the link between genome size and number of genomes, number of genes, which will mark the end of prokaryotic genome discussion. Okay, and then what we will be moving towards is the eukaryotic genome. Okay, so prokaryotic genome is something that we should be able to finish in our next session, and the session after that we will set into our eukaryotic genome discussion. Okay, any queries that you have till this point, you can speak that out. If not, we are good to stop for today.